It's such a pleasure to be here and such a pleasure to be part of this community. I have learned a tremendous amount from MPG, and it's a really honor to give back a little bit by giving one of these primers. So what I'd like to tell you today is about the non-coding part of the genome, the other 99%. So what we have been doing is try to understand how does genetic variation affect human biology. And the field focused a lot on protein coding variants because they have the strongest effects and they are much more likely to be causal when they are associated with disease. The problem is that they often are not the driver of many of the associations that we're finding. So the big surprise of the field over the last 10 years is that non-coding variation is really underlying more than 80% of genetic association results. And I'm going to take you through some examples. So this is my own genome. So you can see here, if you did 23andMe before they were shut down, you would basically have your relationship with your siblings, your ancestry, which you still have, and then this tiny little part here, which basically tells you for every genetic variant that you own, which is associated with a particular trait, what is your predisposition, and specifically for disease-associated variants, are the alleles that you carry increasing or decreasing your risk of a particular trait? So in this particular case, this is the worst part about my own genome. I have an increased predisposition for age-related macular degeneration. So out of 5 genes, 5 loci, I have 3 bad allele combinations and 2 good allele combinations. None of my parents or siblings have that specific combination, and it increases my risk to about 8% when you only have a 6% risk. So the question that you must be wondering is, what do these genetic variants actually do, and what can we do about them? And that's where a lot of the stuff that I'm going to talk to you about comes in. That's where regulatory genomics and epigenomics comes in. So, DNA sequence kind of looks like that. These are actually 3 loci in my genome that are badly associated with a disease. And here's where they sit in genome-wide association studies. So you've heard a lot about GWAS already. And you've seen a lot of these Manhattan plots, which basically tell you for every locus in the genome, what is the association with a particular trait. And you can see here my 3 bad ones and my 2 good ones. Let's zoom in more closely. So ARMS2, TMP3, SIN3, and C2. These are the 3 bad ones. And what do? 2 of them affect protein coding genes. They actually change the amino acid sequence of those genes. And they're involved in the complement complex that helps clear out debris from your retina, which is related to age-related macular degeneration. This one sits 100,000 nucleotides from any protein coding gene. There is absolutely no protein coding alterations in that region by any of the genetic variants that are tagged by this locus. So how do we go about interpreting this locus? And how do we go from these regions of association to the specific mechanism underlying these non-coding variants so that we can eventually have handles for developing drug, so that we can figure out the upstream regulators that target these non-coding variants, the target genes that are affected, and we can start developing drugs either to the upstream regulators or to the downstream target genes, or genome editing tools for the genetic variant itself. So understanding the regulatory circuitry of these regions is in fact what's going to allow us to go from regions and mechanisms to drugs. The second part is that right now we're thinking about all of these genes in complete isolation, but in fact they are part of a regulatory network and they are involved in pathways and systems, and having 2 bad alleles in the same pathway, for example, or in parallel pathways that are involved in the same function could actually be much more detrimental than having either one alone or simply the addition of the 2. So we need to understand the context of how these isolated variants act within the genome. So that's what I'm going to tell you about. So how do we actually understand human variation in human disease? Well, a small prerequisite of understanding that is to actually understand what every nucleotide in the genome does, and not just in one cell type, but in every single cell type of the human body, so that we can predict the effect and the impact of individual mutations at each one of those loci. And again, of course, this is not a small problem either, and it brings in a whole other field to the study of human genetics, which is regulatory genomics and epigenomics, which is what I'm going to be focusing on today. So what is the goal of regulatory genomics and epigenomics? It's basically to understand the regions of gene regulation. So proximal promoter regions is where regulators bind these sequence motifs to turn on transcription and make a product out of a gene, make a protein product eventually. Enhancers are much more dynamic than promoters. They sit much further away from the gene, and they control the tissue-specific and the cell-type specific activity of these genes, while promoters primarily control where transcription is eventually going to start. Okay? So these are the two primary elements that we're going to be talking about throughout the talk: enhancers that are distal and very cell-type specific, and promoters that are proximal and much more stable across cell types. And what we'll try to understand is the regulators that bind these regions, that target them to turn them on and off. Understanding the regions themselves and how to annotate it and their tissue specificity. Understanding the targeting that goes on in the genome. Which transcription factors target what genes, and specifically what regulators target what enhancer regions. So for every one of these nucleotides, we'd like to know who's upstream of it and who's downstream of it, because these enhancers can act up to 1 million nucleotides away. In fact, one of the examples that I'm going to show is an enhancer where a single non-coding variant is affecting two genes, one 500,000 nucleotides away and one 1 million nucleotides away, where again, if we're only looking at the proximal region, we would be completely mistaken as to the mechanism of the gene. So understanding these networks is extremely important. And of course the grammars, the interplay of multiple transcription factors, so that we can ultimately predict the expression levels that are consequent of genetic variations. And I think of these as the building blocks of gene regulatory networks, which we can then use to interpret genetic association studies. So that's how I'm going to organize my talk. First, I'm going to focus on understanding gene regulation. So how can we use evolution, epigenomics, and regulatory genomics for understanding the basic circuitry of the cell? And then I'm going to switch to how do we actually use that in human disease. So first of all, how do we use it to interpret genetic association studies? How do we go beyond individual loci and exploit these regulatory networks across many loci? And how do we then apply that to individual patients to understand the intermediate phenotypes that ultimately give rise to disease? Do we take questions?
Yeah, and if you do, repeat it so that it's recorded.
So feel free to stop me at any time. So this is the menu. Let's now start diving through. So first we're going to talk about evolution. This is basically comparing the human genome to dozens of other mammals that more or less have the same biology. So most of the processes that underlie human biology and even human disease are in fact also present in many of these other mammals. So basically seeing that a nucleotide that's conserved between human and other mammals that is associated with these traits is in fact a very good hint that that nucleotide is very likely to be important. So what do? We basically align dozens of mammals, and then we pinpoint regions that are highly conserved across different mammals. And that allows us to recognize elements that might be important, but not yet the function of these elements. To understand the function of these elements, we can go a step further and not only observe the overall level of conservation of different regions, but actually observe the pattern of change that has happened in these locations. So protein coding genes evolve very differently from non-coding regions, and you can use these signatures to distinguish the two. RNA structures have very specific conservation signatures that preserve the secondary structure through paired substitutions. And microRNAs preserve, in addition, the sequence specificity that allows them to target protein coding transcripts. And then lastly, regulatory motifs are very short and they can move around a lot in the genome, and they, however, have their own phylogenetic conservation in any individual locus that allows you to recognize them, as well as the property that they're conserved in thousands of locations across the genome, which can boost your power for discovering them. So we can use comparative genomics and discover both the regions of conservation as well as the specific classes of conservation, which then give you hints as to the underlying elements. So now we can start increasing the resolution by looking at these blocks of conservation that I showed you in the previous plot. And when you zoom in, you see that some of these blocks are in fact separated further and correspond to individual sequence motifs that are conserved, and in this particular case also bound by the corresponding regulator. And you can use that to basically say, well, if a variant lands here, and it's in perfect LD with my trait, and another variant lands here, I'm much more likely to believe this one because it actually disrupts an element that evolution has minutely preserved through about 60 million years of radiation of the mammals. So you can also use the same approaches within the human genome. You can basically compare the sequence changes between different humans and observe their allele frequencies or the density of SNPs. But of course, you're much less powered here because the mammalian tree is much, much larger than the human tree. So the human branch is this tiny little blip here. And even in that blip, human population itself is a much, much smaller blip. So the power that we have for individual regions is very weak, but we can get a lot of power by aggregating regions. And here you can see an aggregation plot of about 10,000 different instances of the CTCF motif. And you can see that we can distinguish, based on the level of heterozygosity, bound regions from non-bound regions, because the bound regions are actually much more likely to be conserved between different humans than the non-bound regions. So you can see that selection is very actively working in the human genome to preserve the ones that are actually more likely to be functional. And again, when you have multiple SNPs that are associated with a particular trait, and you've heard a lot about GWAS and how linkage disequilibrium is actually often giving you many SNPs, any of which could actually be the causal one driving the association. So a lot of the challenge of interpreting GWAS is figuring out which of these is more likely to be the driver. And when you have one of them which is in fact conserved across species, which disrupts a conserved nucleotide, then you're much more likely to believe that this is actually real. So the second signature that we're going to talk about is epigenomics. So using the packaging of DNA around these nucleosomes that make up chromatin to actually understand the underlying functions that are being encoded in each of these regions. So as you know, DNA is a double helix, and it's probably the most noble molecule of our time. And every 250 nucleotides of DNA are wrapped around a nucleosome. So 147 sit here, and another 50 sit here. So every 200 nucleotides, you basically have another nucleosome. And what's really very exciting about these nucleosomes is that they're not only structural, they don't only hold the DNA together, they're also informational. So you can actually have specific post-translational modifications on the proteins that make up nucleosomes that are known as histones. And these tails of these histone proteins have long stretches of amino acids that are sticking out of the nucleosome, which can actually be modified with methylation marks, acetylation marks, phosphorylation marks, and so on and so forth, which are read by specific regulators that can then act on the chromatin itself. They're also written by other regulators that basically encode specific signals like post-it notes. So if you have a little yellow post-it note or orange post-it note, you're more likely to be an enhancer region. If you have a red post-it note, you're more likely to be a promoter region. If you have a green one, you're more likely to be a transcribe region. And a gray one, you're more likely to be a repressed region. So the way that the single genome that we have gives rise to the diversity, the astounding diversity of cell types that the human body can encode, is in fact through these epigenetic modifications, because in each one of these cell types, a different set of genes is marked for activity, giving rise to the specific cellular and tissue level structures. So what we study in epigenomics are basically those modifications of the DNA. So what are these modifications? So first of all, CPG dinucleotides, so C followed by a G in the 5' to 3' direction in which DNA is read, can actually be methylated which actually represses many regulatory regions and prevents genes from being transcribed. So DNA methylation itself is happening at the level of the DNA. Then you have accessibility, which basically says, are these nucleosomes blocking or not a particular binding site that is now no longer accessible to a regulator? And then the third level is histone modifications. So these post-translation modifications that I mentioned on these tails of the histone proteins are basically extremely diverse. There are more than 100 different modifications that can basically give an astronomical number of combinations of modifications. So I'm going to focus on a small number of those here. So enhancer regions that I mentioned earlier, these orange post-it notes, are basically marked by a single methylation group on the fourth lysine of histone H3. So basically you have H2A, H2B, H3, H4. This is H3 right here. It has a long tail. Lysine at position number 4 can have one methylation mark. So that's H3K4ME1, and that's an enhancer region. H3K27 acetylation is basically the 27th position, has an acetyl group. And DNase basically says that when I try to cut DNase, I'm more likely to cut here because that region is actually accessible. So together, this signature defines enhancer regions. A different signature defines promoter regions. Now you have a trimethyl group at that position, and you have an acetyl group in the 9th lysine rather than the 27th. So again, you have a different signature for promoters. Transcribed regions, yet a different signature, and repressed regions have several different signatures. So you can now use these signatures to start interpreting the genome. Okay? So again, this is a primer, so let me actually take you through how we actually go about measuring these modifications. So when I tell you H3K4ME3 is sitting there, well, how do we actually know that it's sitting there? We know this by actually doing chromatin immunoprecipitation. So what does that mean? That means that I can develop an antibody against that particular modification that recognizes the 27th lysine acetylation property of histone H3. So once I have an antibody, I can then cross-link proteins in DNA, lyse the cell, sonicate, fragment the chromatin through sonication, and then use my antibody to pull down segments that have that particular modification, or that particular regulator, or that particular any kind of property of the DNA. And then when I pull this down, I end up with an enrichment of regions of the DNA that actually had that modification. And what do? I simply sequence the heck out of it. And then I map all the remaining reads onto the genome. And that gives me peaks. That basically says that when I looked for H3K4ME1, the locations that I found it were much more likely to be here and here and here and here, but definitely not here or there. And I can do that for each one of a large number of modifications. So that gives me now these extremely complex maps that basically tell me for very different types of modifications, as well as for DNA accessibility, as well as for RNA transcription and DNA methylation, where are these regions found across the genome? And I can combine all of that information together to interpret the underlying state of the chromatin. So what does that mean? That means that I want to go beyond these single marks. I want to have a single track for every cell type that basically tells me that this is an enhancer region, that's another enhancer region, that's a promoter region, that's a transcribed region, that's a repressed region, and so on and so forth. And notice here the correspondence with the genes. So this particular gene is actually repressed. This particular gene is highly active. It has this long enhancer right there, and a lot of evidence of transcription. So we developed this computational technique for automatically learning combinations of histone modification marks from the primary signal that can de novo discover specific types of chromatin modifications. So you can see here promoter regions are associated with a large number of different combinations, transcribed regions with other combinations, enhancers with other combinations, and so on and so forth. So you can now take different cell types, and that's the beauty of this approach, and summarize them into a single track per cell type. So when you have many different modification tracks per cell, it's very difficult to carry out any analysis. But when you combine them all together, you end up with a very clear map of what is the underlying biology, if you wish, that is reflected in these individual marks, but that is better interpreted in the context of these mark combinations. So we give names to these combinations. We basically call them active promoter, weak promoter, poised promoter, strong and weak enhancer, insulator, transcribed region, polycomb repressed, heterochromatin. But this is the only part where the human intervenes. Everything else is learned automatically by the machine. So we know that this actually gives us an unbiased view of the genome. Given the particular experiments that we have, the algorithm learns automatically what are the underlying sequences and what are the underlying signatures. And you can also, by having this across every cell type, go beyond that and start looking for correlated patterns of activity. So this particular enhancer here is closer to this gene, but it is correlated with the activity of that gene. Every time that enhancer turns on, that gene seems to be turning on. So we can now start using that to link these regions together. And we can do that across hundreds of cell types. So as part of the Roadmap Epigenomics project, we have now profiled more than 100 different adult tissues, embryonic tissues, stem cells, stem cell-derived iPS cells, and primary cells from different regions of the body, and profiled a large number of different modifications for these, for each of these samples. And what that gives us is a picture like this, where for 127 different samples in this particular picture, which we can group into the type of provenance, for example, brain or heart or digestive tissues or immune cells and so on and so forth, we can basically at a glance see how these regions are active across different cell types. So you can now, for example, here see the same PAX5 gene, which was repressed in the example that I showed earlier, IMR90. You can now see that expanded across 127 epigenomes and see that a couple of cell types, in fact, escape this polycomb repression. And they are very actively transcribed. You can see the promoter here, enhancers, and then the transcribe marks. You can see other genes that are very linear specific. So in epithelial cells, this turns on. In heart, digestive tissues, this turns on. In stem cells, this turns on. But it turns off in immune cells and so on and so forth. You can see other genes that are very constitutively active and other genes that are much more patchy in their activity. Again, this is the first time that we've been able to observe the human epigenome at such a diversity of tissues across the human body. Notice also that promoter regions that I mentioned earlier are right next to the gene start are in fact very stable across cell types. You can see here these red streaks coming through. And you can see that enhancer regions, these orange tick marks, are much more dynamic. So we can actually play a game here and take all of the orange regions across not just this region, but across the entire 3 billion bases of the human genome, and stack them side by side, clustering them based on their common activity. So what that gives us is a picture like this, where we've now taken 2.3 million enhancer regions, and we look at their activity across all the cell types. You see that there's a very small minority that are broadly active, and the vast majority are extremely cell type specific. And when you look at the genes that are near these clusters, they're enriched in very specific processes that are associated with the corresponding tissues. So that gives us a very dynamic view of the human genome, which we can actually use to understand its circuitry. Yes, please.
How stable are these? Like, for example, some of them are off all the time, but for ones that go back and forth between tissue types, if you went back again, would you get the same answer?
That's a great question. So the question is, if an enhancer appears to be extremely dynamic, and in my particular view, I see that in heart, it happens to be active. If I sample a heart tissue again from another individual, is that going to be active again? The answer is probably yes. Namely, the primary driver of the epigenomic landscape is really the tissue that something is coming from. The mixture of cell types within that tissue can influence your answer. For specific tissues, whether you've eaten or not, or slept or not, and so on, could influence your answer, because many of these are actually dynamic and they respond to different stimuli from the environment. H3K4ME1 is actually fairly stable. H3K27 acetylation is fairly dynamic, and they're both enhancer marks. So you can play that game to basically look for things that vary a lot and things that vary less. And in the end of the talk, I'll talk a little bit about how these marks vary between individuals, but the short answer is very little. The very small part that varies is extremely important and extremely interesting. We can, make thousands of stories and read a lot and learn a lot from that, but it's still a tiny fraction that varies. The vast majority is driven by the tissue. There was another question? Yeah, please.
How does ChIP-seq perform from paraffin fixed, previously fixed sections?
Yeah, so there's some preservation methods, such as PAX-gene, that actually effectively destroy the chromatin marks. DNA methylation you can still observe, and these marks are correlated with each other. So you can go and profile, for example, DNA methylation through PAX-gene fixed tissue and infer something about the activity of that region across individuals, for example, even if all you have is fixed tissue. For flash frozen, you can absolutely do ChIP-seq. So other questions? Great, so now we're going to exploit these modules that we've learned to actually learn something about the circuitry of the cell. So as I mentioned earlier, in that one example, chromatin state correlation with gene expression patterns can allow you to link enhancers to their target genes. So we do this now using our modules by basically learning modules across a large number of cell types, and then learning gene expression modules across a large number of cell types, and then linking our patterns of enhancer activity to our patterns of gene expression activity. And that allows us to get a much more robust linking between members of each of these clusters than what we would get if we were trying to link each one of 2.3 million enhancers to 200,000 genes. So once we have these links, we can now ask, well, first of all, what cell type was this link discovered in? And start comparing the specific cell type in which something was discovered with lines of evidence from physical interactions of the genome that folds in 3 dimensions through Hi-C or ChIA-PET, which basically tell you what regions are in close proximity in 3 dimensions, either in an unbiased way or through the lens of a particular protein. And you can also compare that with EQTLs, and I believe you've already heard about EQTLs, which are genetic variants whose differences are across individuals are associated with expression differences between individuals. And these actually give you links on the genome. And it turns out those links are very highly predicted by our cell type specific links in the matching cell types, giving us confidence that the links that we're predicting are in fact both real and capture the cell type specificity. But we can go a step beyond that and not only study the correlation between these chromatin states and expression, but also correlation between these chromatin states, the expression of transcription factors, and the binding motif enrichment of these transcription factors in these regulatory regions. So if you try that, you basically take our enhancer modules here and how they're active across different cell types, and then for each one of these modules, find regulatory motifs that are enriched or depleted in each of these cell types, and correlate that with the expression of the corresponding transcription factor. And orange means here a positive correlation, suggesting an activator, and purple here means a negative correlation, suggesting a repressor. And in this particular case, REST, also known as NRSF, is actually very highly enriched in brain, but the corresponding factor is negatively correlated, because NRSF is in fact a transcription factor which is very highly expressed everywhere in the body except brain, and is specifically keeping off brain enhancers from being turned on. So again, you can distinguish repressors from activators, and you can put all of that into a regulatory network that tells you what transcription factors and their corresponding motifs are in fact activating or repressing neighboring tissues across our map here. So that allows you to now go back into these genome-wide association studies and say, well, if I find a motif inside an enriched enhancer, I'm much more likely to believe it's causal than if I find it in some out-of-context setting. So HNF4 is a motif that's enriched in liver, and therefore when I find HNF4 in liver, I'm going to call it a driver instance, and when I find it in brain, I'm going to call it a non-driver instance. This is in context, this is out of context. And now what's really exciting is that if you look at mammalian conservation, you see that the in-content motif instances are much more conserved than the out-of-context motif instances. So evolution is preferentially preserving the ones that are in context. Even though, of course, we didn't tell it anything about that. It's just simply that if you disrupt them, you're much more likely to have a consequence on fitness, and therefore they're excluded by mutations across evolutionary time. And what's really also exciting is that if you look at the relative human constraint, the coverage-corrected relative constraint from the 1,000 Genomes Project, you again see a difference where the out-of-context motif instances are much less seemingly constrained within the human population than the in-context motif instances. So this is, good evidence that what we're predicting to be causal, simply based on the dynamics across different cell types, is in fact supported by evolutionary information. It's also supported by experimental information where we actually test a lot of these sequences by systematic perturbations in a plasmid to basically see if I test that sequence, say 145 nucleotides surrounding this particular motif, with and without a mutation, if they're likely to drive a reporter construct. So in collaboration with Tarzan Mikkelsen, we actually tried this in 2 of the cell types, predicting 3 activators in one, 2 activators in the other, and 1 repressor in each, and went through and tested about 2,000 different enhancers. Here's one such example where we have different barcodes for each of the enhancers, and we're now testing 145 base pairs for the wild-type enhancer, which is active in HepG2, liver, and inactive in this blood cell, and we perturb the sequence of that predicted driver motif in many different ways, and we find that indeed scrambling, removing, or maximally decreasing a single base pair of that motif is in fact abolishing the expression of the reporter gene. Making silent changes does not change it, and making random changes, well, it depends on whether you perturb the motif or not. Again, giving us credence to our regulatory predictions. So we've basically used evolutionary signatures to figure out which variants are more likely to be more important, epigenomic signatures to figure out what cell type they're much more likely to be active in, and regulatory genomics to actually predict the specific motifs that are likely to be active. And now we're going to turn to applying this to interpret human disease. There's a question.
What silent change means?
Oh, absolutely. Thank you very much for saying this. So the question is, what does a silent change mean? So for protein coding regions, we know what a silent change is. A silent change is something that does not alter the amino acid sequence. For non-coding mutations, it's much more difficult to figure out what silent changes are. But when we do know the regulatory motif that binds there, we can actually see that in some locations, I have CAAG, and then either G or T will do at that position. So a silent change is something that changes a T to a G, but preserves the consensus of that motif. Thanks for a great question. Any other questions? Okay, so now for the second part of the talk, I'm going to turn to applying what we've learned about the human genome circuitry and its cell type specific nature to actually start interpreting disease. First, we're going to do that for individual variants, then we're going to do that in the context of networks, and then we're going to look at epigenetic changes across patients. So you've heard about GWAS before. I'm not going to reintroduce it, but basically when you have a genetic variant that's associated with a difference between cases and controls, that tells you that something in that region is in fact driving a difference in biology between cases and controls. But of course, each of these variants is in linkage disequilibrium with many other variants that are co-inherited across the human population. So if I have a particular G at that particular location, I also probably have a T here and an A here and a G here, and therefore about 20 nucleotides that are changing in about a 20 kb interval, I can more or less predict by having that particular variant. That has been a huge advantage for genome-wide association studies because it allows us to map a limited number of variants across individuals and then infer many, many more variants. But the moment you have that association, you're now in trouble, because what gave you the power to only type one variant out of 20 is now a hindrance, because you don't know which out of those 20 is the causal one. And that's where epigenomics can come in. So we can take now all of the variants that are genome-wide significant for different traits and ask how many of those overlap enhancers in specific cell types. So if you look here, in systemic lupus erythematosus, there are 18 genome-wide significant variants. 6 of them land in GM12878 lymphoblastoid enhancers. So you can see here that 6 out of the 18 genome-wide significant variants are in fact annotated as enhancers. Is that significant? Well, if I use a permutation-based approach, or simply a background approach where I basically ask how much more, what would I have expected by chance, I would have expected maybe half of these 18, like 0.1 or 0.5 out of 18 on average, and I find 6. This is a very significant enrichment, which basically gives me a significant FDR. And what's really exciting is that it's actually cell type specific. So different cell types here show different enrichments for different traits. So that allows me to, in a completely unbiased way, figure out what cell types are more likely to be relevant for different traits. And of course, the moment I have that, it doesn't just give me an enrichment, it gives me a way to now probe into the variants that are the more likely to be causal. In this particular case, there's a SNP that's in perfect LD, perfect linkage to disequilibrium, with the typed SNP in the study that actually disrupts an ETS1 motif, and therefore I can make the prediction that perhaps the binding of ETS1 is actually altered in that individual that contains the risk allele, and therefore changes the regulation of these neighboring genes, which is in fact predicted to be linked through correlation analysis, and therefore go from a non-coding association to a pretty precise prediction about what could be underlying that association. So I can predict the regulator, the cell type where it's active, and the target gene, which gets me much more further than simply knowing that there's one of possibly many different variants in that region driving the association. So again, I can take this now across 127 cell types and look at a large number of diseases and traits. And I can predict the most enriched cell type for each of those diseases based on the genome-wide significant variants. And if you look at, for example, attention deficit hyperactivity disorder, you basically see that the highest enrichment is in brain. This is kind of exciting. If you look at PR interval, or repolarization interval, or blood pressure, or aortic root size, the most enriched cell type is in fact heart, which is kind of nice. If you look at a bunch of immune traits, the most enriched cell types are in fact enhancers in immune cell lines. If you look at cholesterol, HDL, or metabolic levels, or LDL, you basically see over and over again an enrichment in liver. So this is pretty exciting. It basically says that if we knew nothing about these traits, we could actually predict what's possibly underlying their biology, and we can use that to now go back into genome-wide association regions, just genome-wide associated regions, and look for the variants that land in the corresponding enhancers, because they are now much more likely to be causal. So, of course, increasing the power of these studies by doubling our cohort size or profiling 10 times more people, we could get at the same answer. But this allows us to now start prioritizing variants even though we may be underpowered. And that's extremely important, because for some diseases, we actually have every variant on the planet. We've got them all. At most, we could double if we looked much harder. So we are limited by the ability to sort of use genetics alone. And that's where epigenetics can actually complement that to basically prioritize specific loci. So we've done this for a bunch of traits in various collaborations, but we've also put up a website that allows people to just go and mine their own genome-wide association study, look for all of the variants in LD, find specific hypotheses, follow these hypotheses up through these regulatory models that we're predicting, figure out who the driver motifs might be, and so on and so forth. So this is pretty exciting, but it doesn't stop there. Because again, if you look at the genome-wide significant variants that I'm using here, they explain only a small fraction of the heritability of each of these traits. We know that there's much more in the genetics than what we're capturing based on the common variants that are genome-wide significant. Moreover, statistical studies that use all common variants that are actually typed by our array suggest that even the common variants that we are typing are capturing additional heritability, additional information about what the genetics tells you about the disease. So these are referred to as missing heritability and hidden heritability. The missing one is the one that might be in rare variants that are currently not typed. And then the hidden one is the one that we've actually profiled already, but we just don't know how to interpret. And this can actually make a big difference for that. So let me illustrate this by looking at our enrichments below the genome-wide significance threshold. Okay? So genome-wide significance threshold is like the first pixel here. And then I can extend this across, say, 2.3 million variants that are either typed or imputed in any particular study. And I can ask, what is the enrichment that I find at different places along the rank list above this expected x-axis where all of these curves are going to eventually land back for different annotations. In this particular case, I'm looking at type 1 diabetes, and I'm ranking all of the SNPs based on their association with type 1 diabetes. And then I look at immune enhancers, and I look at some other cell type. And what I find is that immune enhancers are very strongly enriched at the top of the rank list. And that enrichment continues for tens of thousands of variants. So if you look here, you can see that a large number of immune traits or of immune enhancers are in fact associated and enriched in the genetic variants that are found at pretty non-significant p-values. You know, 10 to the minus 8 is here, which is the genome-wide significance threshold, and then 10 to the minus 5 is around here, 10 to the minus 4 is here. I mean, by here we're at 10 to the minus 3, which many people wouldn't even pay attention to. But now, these enrichments suggest that we can prioritize additional loci when they land in these regions. Okay? We find the same thing with somatic variants associated with cancer. When we start linking every gene to all of the regulatory regions that it is connected to in 3 dimensions through physical proximity, we find that a large number of somatic mutations are recurrently occurring in the non-coding regions sitting under enhancer chromatin states for a large number of genes. So that allows us to now predict driver genes in cancer, even when they don't contain any protein coding alterations, because they contain regulatory variants repeatedly across different individuals. And in one collaboration that was led by Xin-Cheng Wang, as well as Laurie Boyer here at MIT, involving Chris actually on the genetic side, we actually said, can we take some of this sub-threshold variants and actually test them? So we tested a number of genes that were sitting near these sub-threshold variants associated with repolarization duration in heart. And what we found is that the predicted targets of these genes, when targeted, when knocked down by morpholinos in zebrafish, in fact lead to a significant change in repolarization duration. So these are new genes now that we've discovered by prioritizing variants that were sitting in these epigenomic annotations that are enriched, even though they do not meet genome-wide significance. So to discover those de novo, purely based on the genetics, you need much larger cohort sizes. And in fact, in 2 of these cases, the larger cohorts that Chris had actually put together had discovered 2 of our candidates that we had prior to increasing the size of our cohort, suggesting again that this is giving us a lot of leverage to go further. So the last thing I'm going to talk about is how we can actually connect these enhancers to genetic variants, regulators, and targets to get much more insight into the specific mechanisms of enrichment. Why? Because individual cell types are still a very coarse set. There are thousands and tens of thousands of enhancers in any one cell type, only a subset of which are involved in specific processes at any one time. So what we can instead do is start bringing in the regulators to start further narrowing down the associations. And we can do this in an iterative fashion. We can basically link all of the genetic variants to enhancer regions and then use those to prioritize regulators that are likely to be drivers, and then iterate. Basically, once we can predict these regulators, we can use these regulators to further prioritize the enhancers that are likely to be causal, and then use that to refine the regulators, refine the enhancers, and so on and so forth. So we start with genome-wide p-values, a regulatory network, and then a network connecting every region to a SNP. And then we end up with trait-relevant transcription factors, potentially causal SNPs, and predicted target genes. So this is one such example where there are many SNPs in LD, only some of which are in fact multiply targeted across the whole genome, allowing us to now prioritize specific variants and specific gene combinations. We can also start looking at genetic variation specifically in patients, okay? So much like I showed earlier, you can actually study how cases and controls differ in terms of their genetics. You can also do that in the study of an expression trait. So basically, you can look at high expressed genes or low expressed genes at a particular locus, and then look for genetic variants that are associated with that. Or you could actually associate directly epigenetic modifications in these individuals with the traits of those individuals. And what that allows us to do is actually start bridging this gap between the genetic variant and the disease. So we can basically predict specific tissues in which the disease might be acting through our enrichment, and actually look for specific gene expression changes in those tissues that might be intermediates to the disease, and then specifically endophenotypes that might be associated with the disease that could, again, help bridge that gap, and regulatory variation at the epigenetic level specifically for those variants. So that allows us to now start making much more specific predictions about the intermediate phenotypes and start addressing one of the challenges of genome-wide association studies, which is that the traits are extremely complex. So any one variant may have a very small effect at the disease level, but a very strong effect in the specific tissue, the specific regulator, the specific gene expression level. And the more we bridge this gap, the stronger the effects that we're going to find are, and that allows us to actually make much more specific predictions. So this is one example from Jonathan Pritchard and Yoav Gilad's group, where they basically looked at several different histone modification patterns and how they are affected by a single variant here, where the homozygotes for A or the homozygotes for G show very different levels of activity, and the heterozygotes are somewhere in between. So we can now use that in the context of disease. So in collaboration with Phil DeJager and David Bennett, and as part of the Roadmap cohort, we basically profiled methylation differences in the dorsolateral prefrontal cortex of 700 individuals and looked at how genotype changes, epigenome changes, and phenotypic changes are related to each other. So here's a glimpse at the data. You basically have the chromatin state annotations across several regions of the brain and a lot of non-brain regions from the Roadmap Epigenomics project. The methylation levels using a 450 lumina array across 708 individuals, and then the genotype across 586 of these individuals at the same location. So now we can start looking for strong associations where this vector is highly correlated with that vector and highly correlated with disease. In any of these studies, you need to control for a lot of covariates that are leading to global changes rather than specific changes, and we control for a lot of them. That increases our signal. And that allows us to now predict more than 50,000 methyl QTLs. When you do a GWAS, you have 10 things above that genome-wide significance threshold, or 100 if you're lucky. If you if you overlay now the methyl QTL annotations for all of the probes across the genome-wide significance after Bonferroni correction is that red line down here, and we have 50,000 variants that are very highly predictive of methylation levels. Even down to here, you can basically see very significant changes. These are very highly concentrated near the target CPG that they're affecting. and the power to discover them increases almost linearly with the number of individuals, suggesting again that bigger cohorts will make a very big difference. But again, this number is astronomical here compared to GWAS, and the reason, I believe, is because we're looking at a much more simple trait, which is much more directly affected by genetic variation. So what's really exciting is that if you now start studying the chromatin state annotations of the corresponding methylation probes, you see that enhancers are much more variable between individuals than promoter regions. And they're also much more likely to be associated with QTLs than promoter regions, suggesting that genetic variation can affect enhancers much more than promoters. And also, they're much more predictive of Alzheimer's status than promoter regions. So enhancers are here, all genome methylation is here, and promoters are here, suggesting that focusing specifically on methylation changes in enhancers is actually very informative. And then when you compare that to using ApoE4 alone or SNPs alone, you're actually doing much, much better, as you would expect. Of course, not all of this is causal. Many of them are consequences of the disease. So we can actually ask, are genetic variants that are associated with Alzheimer's disease below genome-wide significance threshold, so we're now taking the top 1,000 variants, are they enriched in the regions that show these methylation differences? And the answer is remarkable. Indeed, across tens of thousands of probes that are associated with disease, genetic variants are also colocalizing in the same regions, suggesting again that epigenetic changes have the power to reveal regions that are actually causal.
Probably about 2 minutes.
Yeah, perfect. And if you look at the association with Alzheimer's, you find that enhancers are at the top of the list. If you look at the association with age, you find that polycomb targets are at the top of the list, suggesting that different chromatin states are in fact underlying different phenotypic associations. And when you look at the genes that are near these epigenetic changes, you find that they're very strongly enriched in specific pathways that are relevant to Alzheimer's disease. So what I told you about is how we can understand regulatory circuitry through the use of comparative genomics, epigenomics, and regulatory genomics. And we can use all that to apply it to interpret disease through understanding genetic associations, linking together these variants into networks, and then applying that directly to observe epigenetic changes in patients. So I'll finish by thanking the folks who are doing the work. So Anshu Kuldaji, Matt Eaton, Abhishek Sarkar, Gerald Kwon, Luke Ward, Jason Ernst, Wouter Milliman. And this is part of the Epigenomics Roadmap project, which is an outstanding collaboration. And again, this is a brief summary. We can characterize the epigenomic landscape, use the activity patterns, modules, and networks to link these together, and then use that to identify disease-relevant tissues and regulators, and to mechanistically dissect disease-associated loci. We can predict functional roles of weakly associated variants that are not reaching genome-wide significance, and use that even for somatic mutations in cancer. And then lastly, study intermediate phenotypes in the context of Alzheimer's. So I'll stop there, and thank you all for your attention.